Hello everyone. Welcome to the new session of Microorganism, Bacteria Part 1. So in this session we are going to study about microorganism, and the first microorganism we are taking, that is bacteria. Okay. So structure and function of bacterial cell. So let us first discuss about the structure of the bacterial cell. A bacterial cell have essential structural components. So they require some structural component. What are those? Cell wall, cytoplasm, membrane, intracytoplasm structure, and cell surface appendages like capsule, flagella, fimbriae, and also spore. Okay. So biochemical compositions of these structure are macromolecules. So we are having two types of biomolecules, macromolecules and micromolecules. So all the biochemical composition of these structure which are present in the bacterial cell are macromolecules category, and they are arranged or sequenced in primary structure of molecule in which the subunits are put together, such as structure and function of bacterial cell. In this category, DNA and RNA. So they are the nucleotides. The macromolecule here is the nucleotides, and this contain DNA and RNA. Next is amino acids. So amino acids are the building block for the protein. Okay. So this is again our macromolecules. Phospholipids. Phospholipids form the fatty acids. Okay. Polysaccharide, the sugar, it comes under the sugar. So these are the structural component of the bacterial cell. Okay, so let us discuss about the different structure which is present in the bacterial cell. So starting from the outer side, we are having cell envelope. Very, very important, cell envelope. So the bacterial cell consists of cell envelope, cytoplasm, nucleoid, plasmid, inclusion bodies, flagella, pili, and fimbriae. So these are all different structure which are present in the bacterial cell. The number one is cell envelope. So let us discuss about this cell envelope. Cell envelope, it is the outer covering of the protoplasm of the bacterial cell, and it is having three components: glycocalyx, cell wall, and cell membrane. So if we see the outer structure of the bacterial cell, we are having a layer that is called as glycocalyx. Okay. And then comes the cell wall, and then unit membrane, that is the plasma membrane. So this is glycocalyx, this is cell wall, and this is plasma membrane. This is also called cell membrane, right? Now, all these three parts, that is the glycocalyx, cell wall, and plasma membrane, they form cell envelope. Okay. So it has three components: glycocalyx, cell wall, and cell membrane or the plasma membrane. The cell envelope consists of tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall, and then the plasma membrane. Just now we discussed about this. Okay. So although each layer of the envelope perform distinct function, so we are having distinct function for the glycocalyx, for the cell wall, and the plasma membrane, right? But they act together as a single protective unit, and that is why we got this name, cell envelope. All the three components of the cell envelope is having different function. Glycocalyx is having different function, cell wall is having different function, plasma membrane is having different function, but unitedly they perform a distinct function, and that is why it got its name, cell envelope. Okay, cell envelope and its modification. We are having some sort of modification in the cell envelope. Let us see what are those modifications. Bacteria can be classified into two groups, that is gram positive and gram negative, right? So on the basis of the difference in the cell envelope and the manner in which they respond to the staining procedure developed by the Graham. Okay, so Graham was the name of the scientist who developed a stain, and when we stain with this particular stain that is developed by the Graham, okay, either they attain that stain or they do not attain the stain, right? So those that take up the Graham stain are called Gram positive, and the other that do not take that stain, okay, they are called as Gram negative bacteria. So the bacteria are first of all classified in these two groups. Glycocalyx differ in the composition and the thickness among the different bacteria. So the different bacteria is having different composition of this glycocalyx, and their thickness also varies from bacteria to bacteria. Okay, so it could be loose sheath called slime layer. Sometimes it is this glycocalyx layer is called as slime layer when the sheath is loose, okay, while in others it may be thick and tough, and that is called as capsule. Okay, so here the capsule layer is quite thick. It is not a thin layer like loose sheath that is present when then it is called as slime layer, but when it is a thick and tough layer it is called as capsule. But both are glycocalyx, a type of glycocalyx. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from the bursting or collapsing. So cell wall is very, very important, and they provide a certain shape to the bacteria, and it also prevent them from the bursting or collapsing. The plasma membrane is selectively permeable in the nature and interact with the outside world. So this is also very important layer of the cell envelope, that is the plasma membrane. This membrane is similar structurally to that of eukaryotes. So there is not much difference between the cell membrane or the plasma membrane of prokaryotes or the eukaryotes. Means the bacterial cell plasma membrane and any eukaryote plasma membrane is sort of same. A special membranous structure is the mesosome, which is formed by the extension of the plasma membrane into the cell. Now, there is some difference at some places of the plasma membrane. What happened? The plasma membrane invaginate and form a structure which is called as mesosome. So such a structure are not present in the eukaryotes. It is present only in the prokaryotes, like for example bacterial cell. The glycocalyx is the layer of carbohydrates. Generally it is made up of carbohydrate, that is sugar, that surround the outer membrane of some bacterial cell. It is gel-like substance composed of polysaccharides, glycoproteins and glycolipids. Okay, so the glycocalyx, that is in the two form, slime layer or tough, thick, capsule. Tough, thick or capsule. Either it is present in the slime layer form, thin, and capsule, tough and thick layer. But all these, either it is a slime layer or capsule, it is composed of polysaccharides, glycoproteins or glycolipids. Okay, so all these three substances are present in the glycocalyx layer. Cell wall. The bacteria are surrounding by a rigid cell wall. So they are having rigid cell wall. You can see here this yellow one, right? And this one, peach color, that is the capsule. Cell wall and plasma membrane, this form cell envelope. Okay, so the bacteria are surrounded by the rigid cell wall. The principal structural component of the cell wall is peptidoglycan. So here this cell wall is made up of peptidoglycan. Cell wall consists of a polymer of two sugar derivatives, N-acetylglucosamine and N-acetylmuramic acid, NAM, okay, and NAG, linked by the short chains of the amino acid, that is the peptide. These molecules is a type of peptidoglycan called murine. So N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM, NAG and NAM. These are the two sugar derivatives that are present in the polymer form and they constitute the cell wall. Okay. Now this peptidoglycan, which joins the chain, they form the short chain like a structure of amino acids. Okay, that is the peptidoglycan called murine, is a complex of a polysaccharide and polypeptide. So these are the complex structure of the polysaccharide and polypeptide. That is the peptidoglycan. Most bacteria are classified according to the reaction of Gram stain. Okay, we have already discussed about the Gram stain. That was the name of the scientist who discovered this particular stain, right? And when the Gram stain is stained with the bacteria, so the components of the cell wall into major groups, gram positive and gram negative bacteria, based on staining property. So what we are considering, why some of the bacterial cell attain the stain while other do not, because of the component of their cell wall. So cell wall is the responsible for getting stain or not getting any stain. That is in the case of gram negative bacteria. So the gram stain developed in 1884 by the Christian Gram, the most widely employed in the bacteriology lab. So this is nowadays also we used to know whether the bacteria is a gram positive and gram negative. So it also give you just by staining. Now you know the components. What are the components of the cell wall of the bacteria is having? So this layer is a thick gram positive. So we are going to discuss about the gram positive bacteria cell wall. Okay. So this layer is a thick one, thick in gram positive bacteria, constituting 50 to 80 nm of cell wall and responsible for the rigidity of the cell wall and retention of the crystal violet dye. So this is the dye that is crystal violet in color and during the gram stain procedure. So now you know the width of the cell wall, that is 50 to 80 nm. Thick cell wall in the case of gram positive. Okay, so the large amount of PG, peptidoglycan, make gram positive bacteria susceptible to the antibiotics penicillin that inhibit the cell wall synthesis. So large amount of peptidoglycan that makes gram positive bacteria susceptible to antibiotics, okay, that can inhibit the cell wall synthesis. Teichoic acid and the thin layer of lipid is also present. Large amount of peptidoglycan make gram positive bacteria susceptible to the antibiotics, that is the penicillin, one of the example, penicillin that inhibit the cell wall synthesis. And teichoic acid is a thin layer of the lipid that is also present here. Gram positive bacteria is having teichoic acid as a thin layer of lipid. So they are having a thin layer of lipid in the form of teichoic acid also, right? A large amount of peptidoglycan make gram positive bacteria susceptible to the antibiotics like penicillin, right? So they are susceptible to the antibiotics like penicillin because they are having large amount of peptidoglycan and that inhibit the cell wall synthesis, right? Inner layer of the peptidoglycan. This layer is thin, constituting 5 to 10 nm of the cell wall, which cannot retain the crystal violet stain. So this particular part peptidoglycan, that is 5 to 10 nm, they do not retain this crystal violet stain. Outer layer of the lipopolysaccharide LPS containing of the lipid A, that is the endotoxin and the polysaccharide. So this is the component of the outer layer that is made up of lipopolysaccharides. Periplasmic space is present between the inner and the outer layer. So it is filled with the gel and it is crossed by the lipoprotein molecules to link the peptidoglycan layer and LPS layer, and no teichoic acid is present in this particular space. So this is the periplasmic space present between the inner and the outer layer of the wall. So here we can see the gram-positive bacteria. This is the peptidoglycan. This is the peptidoglycan layer. Okay. And it is having the periplasmic space that is having the lipoprotein. And this is the cell membrane. Okay. So now here we are having the gram-negative bacteria. Here we are having outer membrane. Then we are having the peptidoglycan. Okay. And now you can see the length of this peptidoglycan, that is very less. Okay. And the periplasmic space is here, that is filled up with the lipoprotein. And the cell membrane is in both the case is same. Okay. So here we are having in the gram-negative bacteria porine also. Porine, that is the pore in the outer membrane. Right. And the lipopolysaccharides are also attached to the outer surface. So what is the function of the cell wall? Protection the inner structure. It provides protection to the inner structure. It maintains the shape of the bacterial cell. Okay. So it provides a proper shape to the bacterial cell. Contain component which is toxic to the host cell. Okay. And it plays a role in cell division. So these are some of the important function of the cell wall. Example of cell wall deficient bacteria. So there are some bacteria which do not contain the cell wall. So the one is Mycoplasma. This is naturally deficient in the cell wall. No cell wall is present. So it doesn't have any proper shape. Okay. So Mycoplasma is pleuromorphic shape. It can change its shape and do not affected by the penicillin treatment, right? Some of the bacteria under the certain condition are fail in the synthesis of the cell wall when the cell is subjected to penicillin, that is the drug or the lysozyme. So this is sometimes some of the bacteria do not form the cell wall. Next is the cell membrane. Okay, so cell membrane is composed of two layers of lipid. The lipid linked to proteins and to polysaccharides. So we all also know that two lipid layer or the bilayer is present. Okay, and some linked proteins are present. Okay, it is located under the cell wall. Where the position of the cell membrane, that is located under the cell wall. Gram-negative bacteria have inner and outer membrane, whereas gram-positive bacteria have only inner cell membrane. So this is the difference. The space between the inner and outer membrane called periplasmic space. So this is the periplasmic space present between outer cell membrane of the gram-negative bacteria is composed of lipopolysaccharides and the lipoproteins, and LPS act as an endotoxin. Function of the cell membrane. It control on inflow of the metabolite. So whatever the metabolite is coming inside the cell, that is controlled by the cell membrane. Okay, from the cell on the active transport of the molecules into the cell because it has selective permeable. So some of the important metabolite that has to come inside the cell. Okay, so they are taken actively. They do not depend upon the concentration gradient, right? So they are selectively permeable. Energy generation by the oxidative phosphorylation. So the cell membrane or the plasma membrane of the bacteria is also concerned for the energy production that take part in the oxidative phosphorylation reaction, secretion of enzymes and toxic material or the toxic chemical, synthesis of the precursors of the cell wall. So what are the precursors of cell wall? Okay, that also get synthesized by the cell membrane and have important role in the synthesis of cell wall. So this is all about this session. We will have one more session on the bacteria. Okay, thank you very much.